Alrighty, so today's exploration, for today we have Hanna, who is a neuroscientist and a postdoc in neuroscience, and she will be talking about how humans and animals can be trained by using repeating stimuli. So I will now hand it over to Hanna. Take it away.
Everything looks good? Okay, thanks. So, like Claire said, I'm Hanna and I'm a postdoctoral associate in the Pictorial Lab at Yale. I just started about a month ago, so I'm very excited to be here and talk about some of the research that I did in graduate school. So, what I really got excited and interested in studying was what happens to how the activity of brain cells change after you learn something new. And once you learn something new, you usually form a memory and brain cells change. So that's what I studied in graduate school. But before I talk to you a little bit more about my research, I wanted to start talking about my pathway to science, what excited me about neuroscience, how I got here, and what I'm doing. So I was born and raised in Karachi, a city in Pakistan in South Asia, right here on the map. It's a very highly populated city. Over here, you can see in this picture, lots and lots of traffic, lots of people. So currently the population is about 17 million, about twice the amount of a pretty big city in the US, which is New York City. So lots and lots of people. And currently, it gets pretty hot in the city I grew up in. So currently in the summers it's between 110 degrees to 115 degrees. And I went to middle school, high school in Karachi, and I was always really excited about science. I loved biology and chemistry, and usually the common trajectory or pathway is to go to med school after you finish high school, and that's what I thought I was going to do all the way up to my junior year in high school, where I realized med school wasn't an option for me and I wasn't that excited about it. So I talked to my college counselor, and she, who went to a small liberal arts school in the US, suggested that I should try, applying to some schools in the US, see what I really was excited about. I had no clue about neuroscience or psychology or how research happens in a lab. So at 18 years of age, I flew from Pakistan to a very small town called Worcester in Ohio. And I went to my undergrad at the College of Worcester, and this was very different than what I was used to. The population was a lot smaller, just 27,000 people, and I had a really fun time in college, and I experienced lots of new things. One of them was I saw snow for the first time. So that's a picture of me in my first year in college, and I'm very excited because I've seen snow for the first time, and that's my friend from Jamaica who's also seeing snow for the first time. So as you can see, both of us are looking ecstatic when, by the falling ice. And the other thing I also experienced was I learned that about the brain. So I'd never taken psychology or neuroscience classes in the past, and I started taking them, and I got really excited and I really enjoyed it. So I majored in neuroscience as an undergraduate, and I learned about this rather grumpy looking guy named Yvonne Pavlov. And he's grumpy, but he's really smart, and he's extremely well known in the field of psychology right now. But what's interesting is he didn't start off being a psychologist at all. He was a Russian scientist, mostly interested in studying the digestive system of dogs, and he ended up being I'm sorry, he ended up being a he ended up getting very well known in psychology, which is a lot of times what happens in science is you start off in a certain way and you end up somewhere else. So the reason he became very famous because he just accidentally discovered classical conditioning, which is a form of learning. So what is classical conditioning? And before I start talking about that, um, if you guys can put it in the chat if you like. Do any of you guys have a dog, especially a big dog at home, or you've seen a dog, a friend's or a neighbor's? And my second question is, if you do have one or seen one, what happens when they see food? It's like a very obvious reaction. Mouth waters, exactly. Don't give away any answers. No spoilers, please. They're excited, that's true. They do start wagging their tail. Drools, yep, drooling, beautiful. Okay, so these are great answers, and they're all correct answers, but the one answer that I've been uh, wanting to look for is that they drool. So basically, like you guys said very well, that a dog sees food, or if they see you eating food, they drool, and that's pretty common. That's something that like all of you have mentioned, happens. And what's also pretty common or not surprising is that a dog is not going to drool when they see something like a bell or a flashlight. But in some instances, what Pavlov saw, what he accidentally discovered, was that he could see that dogs could drool to a ringing bell in some instances. And how did that happen? So Pavlov had technicians because he probably had a lab, and the technicians would bring food for the dogs, and they would ring the bell of his lab or his house, wherever the dogs were. And every time the technicians would be ringing the bell, they would be bringing food, and they did that over a course of repeatedly every day. And in that process, the dogs started learning that there was some link between the ringing of the bell and the food that they were getting. And they were drooling at this time, right? So they formed an associative memory, which sounds more complicated, but basically it means is that they were able to associate or link the ringing of the bell with food that, of course, they were anticipating or like. We all like food, right? To the point now, what Pavlov discovered is that now, when after this repeated presentations of the bell and food kept happening, as soon as the bell rang, and even when there was no food, they saw that the dogs were salivating or drooling because the bell ringing made them think of the food, right? So now the ringing of the bell meant that they were going to get food and they're going to be hungry. And this was a test that they could see that a new memory has formed because the dogs have now learned that the bell means food. So this is what's classical conditioning. And believe it or not, we are, like Pavlov's dogs, very much influenced by classical conditioning on our day-to-day lives. So let me give you one example. Like anybody else, I'm obsessed with ice cream. I can eat ice cream any time of the day, any season. It doesn't matter if it's cold or hot. I love ice cream. And the other day I was walking home from work and I heard the music from the ice cream truck at a distance, right? I couldn't see the music truck. I couldn't see any ice cream. I just heard the music. And immediately my mouth started watering and I started getting hungry, right? Even when there was no ice cream, which is basically an example of classical, like I have been classically conditioned at some point in my life, probably when I was a child, when I heard the music and the ice cream music made me, or the music from the ice cream truck made me associated to the, deliciousness of the ice cream. Now, another example, it's not as great as the ice cream one, but it's important. It happened not to me, but to a friend. So during when we were in college, my friends and I would go to a sushi restaurant, and we loved the sushi restaurant. We would always go to it, and we would order these platters of sushi, and share from it, try different things. One day we went, and we did the same thing. We ordered multiple platters of sushi, and we all shared. And a few hours later, one of my friends got pretty sick. And it wasn't from the sushi because none of us were affected by it. She got sick from it. Probably she caught a bug or something else, and she wasn't able to eat sushi after that. She was ruined for life because she made a pretty strong association or link with the taste of sushi and her getting sick a few hours later. So, um, classical conditioning also helps us survive, and because it helps us avoid some sort of harmful foods that could be poisonous for us and could hurt us. So overall, um, like I just with some of the examples, classical conditioning plays a pretty big role in how it influences our behavior and helps us survive. So I will move on to the breakout room question. Um, can you recall a time when you were a child, or more recently, even, or whenever, when you can think of an example of classical conditioning that has affected your behavior? If you can't think of it yourself, maybe a friend or a family member, or even, the pet example, like Pavlov's dogs. If you have a dog and you've seen that they've been influenced by classical conditioning, and you can discuss. And I'm excited to hear some of the examples you come up with. But yeah, it was some really good examples of the breakout rooms about classical conditioning. So hopefully this gave you a little bit of a chance to think about how your behavior can sometimes be influenced without you even knowing about it or without you even realizing it. So thanks for sharing your examples. That was fun. So classical conditioning is become or has been for a while very useful to a lot of researchers because it's a pretty simple task for a lot of animals to learn, and including humans. And you can then see what happens to different parts of the brain or to brain cells after a memory is formed. And another more interesting and exciting aspect of research in the memory and psychology field has been looking at how males and females might learn a task differently, maybe, or how males might form memories slightly differently or process memories slightly differently than females. So classical conditioning is again used to see if there are any differences in behavior, and as well as if differences in brain activity. So after learning quite a bit about Yvonne Pavlov's work in undergraduate, I was excited to then work and understand in graduate school what happens to brain cells, or what happens in the brain, like how the brain changes after you learn a certain thing and form a memory. So I was a graduate student at the University of Wisconsin in Milwaukee, and I basically was first interested in looking at what are some, or like knowing what are some of the brain regions that are even involved in memories. Is there just one brain region? Are there multiple regions? And the answer that I found was that there are several brain regions over here. And interesting, and what is exciting is that a lot of the brain regions are similar across different species like humans, dogs, or rodents such as mice and rats. So all of these colored structures that you're seeing over here in blue and green and orange, those are brain structures that are involved or participate in some sort of when a learning event or a new memory is formed. So I was most interested in learning about one particular region, which is red here in humans, in this structure here, and the red up here in rodents. And it was called the retrospinal cortex. And out of a lot of these brain regions, this brain region hadn't been studied a lot. So it was exciting for me. I'm like, there's been a little bit of research done, but I want to know more about what happens to this brain region after animals learn a task and form a new memory. So my primary focus, or what I really was interested in knowing, or was measuring activity in this specific brain region, in the brain cells of this brain region, which is called the retrospinal cortex. So, any ideas what are brain cells called? It's got a specific name. I thought I saw someone posted earlier on. You can put it in the chat. So, yep, they're called neurons. Yep, exactly. With axons and dendrites, that's perfect. Yep, neurons is correct. Wow. Okay, well, you guys know a lot more than I did before. So, yep, these are neurons. You can't really see an axon here, but this structure is called the cell body, or the soma of the neuron. And these branches that are leaving the cell body, or the soma, are called dendrites. So I used this pretty big fancy machine, and I used the microscope on it to really zoom in and focus on these tiny neurons, or brain cells, that you can't really see with the naked eye, obviously. So you use a 40x, or a pretty zoomed in microscope lens, to really look into and try to visualize the neurons. And then I was able to see, once I could visualize or look at these neurons, I wanted to see how does activity or information pass through these neurons? What happens to the neurons when they get excited? So neurons are, think of it as an electrical cable. One neuron is connected to another neuron pretty closely, and lots of different neurons are connected in this web with each other, and they communicate with each other through electrical charges or electricity. So when a neuron is agitated or stimulated, so anything can agitate a neuron or stimulate a neuron. For example, a prick on your finger, you putting your hand on a hot stove, a friend squeezing your hand, or if you have learned something new and have formed a memory, neurons are going to get excited, and with that, they're going to push electricity to the next neuron, and that's how they communicate with each other. And that could happen from one brain region to the next, that is pretty further apart or close together. So I've already explained that neurons move through electrical charges, but unlike a wire, electricity just doesn't follow in the most straightforward way. It's slightly different. So these blips that you're seeing, this is when a neuron is electrically charged. And when it gets charged, it basically starts spiking, right? And this happens within milliseconds, like very, very quickly. Like if you look at the x-axis here, this spike, or what is called an action potential, occurs within a millisecond. And on the y-axis is basically change in electrical charge that leads to this spiking happening. So this spike or this action potential happens, but how does this electrical transfer happen, or how is this movement of electricity that makes this action potential or spike happen? So on the skin of the neuron, there are these gates called ion channels that open and close and allow charged particles or charged atoms to enter or exit the neuron. So, it will allow some charged ions to come inside the neuron, which can basically give its action potential, and then it will allow some ions to leave the neuron to bring it back to its rest. So just to recap all that information I just explained, a neuron is almost like an electrical wire, and electricity flows from one neuron to the next to the next. That's how neurons communicate with each other within one brain. Anything can get a neuron excited. any type of stimulation such as a light flashing in your eye, someone holding your hand or squeezing your hand, some like you learning a new event, like a new task and forming a memory, and that's what's going to agitate and excite the neuron, which will then push that information through the neuron onto the next, and that's how they all will communicate with each other in the form of these action potentials or spikes. So, like I said, a learning event or a memory can lead to the spiking of these neurons. And what happens is, so an example of a memory, just to go back to Pavlov's dog's example, or some even of the examples that you were thinking about, because you learned something new and you formed a memory. And in this case, when Pavlov's dogs were learning, or were in the process of learning, they were learning that the tone was associated or connected or linked to food that made them hungry or drool. So in the event now that after they've learned that the tone is linked to the food, just the tone itself makes them think about the food, right? So before before the dogs didn't learn this, before this learning event didn't happen, the bell didn't mean anything to them, right? They didn't learn anything, they didn't form a memory, and when we see animals that didn't learn anything, we only saw one action potential or spike. And when we saw that the animal, after the animals were classical conditioned, a task and they formed a memory, we saw many different spikes and like a larger number of spikes. I mean, so this pattern or trend was seen in many of the brain regions that are involved in memory formation, right? And we saw the same thing, that one action potential before a memory was formed, several more action potentials after a memory was formed. But I wanted to see what happens in my favorite brain region called the retrospinal cortex. Does the same thing happen, or does something different happen? So that is what, so the retrospinal cortex, this is the slice of a brain. And over here is the retrospinal cortex, this area underneath the white lines. And what I saw actually, that it's involved in memory formation. And over here, I saw some action potentials before there was no classical conditioning or no learning, or before a memory was formed. But what was really surprising to me was that instead of the number of action potentials or spikes increasing, I saw a decrease. So instead, I was expecting the neurons to get more excited after a memory was formed, more agitated, but actually the opposite happened, right? Which was like very surprising and interesting, and like basically a good lesson for science, just like Pavlov, who started studying the digestive system and ended up in the brain. Science can lead you to lots of different paths, like you enter with a certain question, expecting a certain answer back, and you see something completely different, and basically you keep asking questions and trying to get answers for them. So this pattern here, when neurons in the retrospinal cortex were clocking before, after a memory is formed, these results were in the male brain. So I wanted to see, what does the same thing happen in the female brain or something different happen? Because other research has shown that sometimes males and females might process a memory slightly differently or very differently or not at all. So again, I'm just showing this again to you, that the number of action potentials or spikes significant or very much are reduced after a memory is formed in males, but nothing happens in females. So the number of action potentials remain the same. Now, this doesn't mean that females can't process a memory as the same way as males do. It just means that maybe there's a different brain region that is more, that works more, or talks less in females compared to males. And these are some more questions that are basically built up on my research that maybe other people might work on in the future, or I could work on the future as well. We're looking at which other brain region are in, what other brain region might be involved in females versus males. So In conclusion, I learned or did in graduate school was looking at that different brain regions don't process a memory the same way. They might respond or get activated differently. Some brain regions might talk a lot more. Some brain regions, like the retrospinal, talk a lot less. And males and females might not be using the same brain regions to after they learn a certain task or form a memory. And so I am very passionate about studying memory and what happens, how it influences behavior, how it changes the brain. So as you know, at Yale right now, I want to continue to study this similar thing, but now I'm looking at different types of memory. So, any questions?
Hi, Hannah. Thanks so much for a great presentation. We've been taking a look at the chat throughout your presentation, and I will tell you some of the questions that the students have. First of all, everyone, I can speak for myself and I can speak for them. Everyone thought it was a very interesting talk. In the breakout rooms, we were all very, very engaged. So thank you. So thanks for such an engaging presentation.
Thanks, Deandra.
So the first question that some students had was, how did Pavlov accidentally discover it? So, like, what was the process after he accidentally discovered it? Did he notice it and then tell people in his lab about it? How did the word get out after he discovered it?
I think that's a good question, and I don't really know the correct answer. I think this happened a long time ago. I think in the early 1900s. So, like I said, and I think it was just like something he observed, and you know, as a scientist, he was a scientist, just not interested in looking at the brain, and he penned it down. He's like, this is an observation, and the other part of science is repeating it. So he repeated it multiple times, and he kept seeing the same thing, and he's like, this is a reliable this is a reliable observation, and I think he wrote it down, published a paper. So yeah, you can actually, if you look at it or Google it, you can find the original works of Pavlov. I think his paper was in the 1920s or something like that, if I'm not wrong.
Wow, well, that just shows everyone that you could already be a scientist right now if you're just paying attention to what's going around you.
100%. That's how it happens. I mean, even with my own research, I didn't expect to see what I saw. And then, you pen it down, write a lot, you write a lot, you have a lot of notebooks, as many scientists here can relate to. You have lots and lots of notebooks, and you write down very, very detailed notes about, your observations, and just repeating it and seeing that you're reliably seeing similar results.
Yeah. And so then we have another question in the chat about your observations. So based on your data, how do you know a memory was formed between your two measurements?
Very good. That's a very good question. So the way you know a memory is that, like, the test is when you just play the bell and you see the dog drool, right? So if you're doing if you don't see them drool, then they haven't made that association with the bell. And sometimes that can happen, very rarely, but it can. And then so like I said, the test is just the bell. In the lab, that's what we call it, the test. We call it a retrieval test, right? Because you just want to see if that memory has been retrieved or it's coming back again.
Okay. Thanks so much for clarifying that.
Yeah, that's a great question.
And we had another question just now about, I guess, the difference or the similarity between memories and reflexes. So are reflexes a kind of classical conditioning?
Yes, that's a really excellent question as well. Your body is very tied to your brain. So the mouth watering, or the feeling of sickness, those are reflexes, but they are in response to that memory that has been formed. So if you don't have that memory, you're not going to have those reflexes. In some instances, there are other reflexes, but in the classical conditioning example.
Okay, that makes sense. So first the memory, then the reflex. So it's all part of the same process.
Yes, 100%.
Great, thank you. We have another question in the chat. What motivates you to study all this, or what motivated you to study all this before?
So I was very interested in learning a memory, like I said, and also sometimes, I was like it plays a huge role in our lives, how that memory is formed. It makes us who we are. Memories, you remember your birthday in childhood, and it really influences what happens in your future. It influences your future actions, what you're doing presently. We're always making memories, and we're learning something new. And I think what was exciting to me was how many ways our behavior is influenced even without us knowing about it. And then I was interested, and then further on, like the looking at males versus females was very interesting to me, because until 2014, the National Institute of Health didn't require you to even study females. You could get away with just studying males. So more and more research came about how females can be more different than males sometimes, or not at all, and that was very interesting to me too.
That's a really good point. We had another observation mostly, but also I guess there's a question sort of in here about some of the pictures that you had showed about the brain. So I think it was on slide 19 here. Yes, yes. So a lot of people were interested in the blue photo and what it really showed. A lot of them thought it looked like an angry face. So can you explain a little bit of what's going on there?
Yeah, so actually I didn't know you guys were using the seahorse example. So this is another brain region called the hippocampus, which is involved in forming memories. And actually the hippocampus means the seahorse. So the blue is basically a dye that we add because, again, we want to get a very clear picture of the brain. And, by the time we make slices, it's very transparent. You can't really get a nice picture of it to show people. So the blue is basically a dye to make it look more clearer, to give it some sort of, so you can visualize it well. So over here, like I said, this is a brain slice, and this is the hippocampus, which is also involved in forming memories, but not it isn't my. This has been really studied a lot by lots and lots of researchers. In the field of learning and memory and psychology and neuroscience. And right above it is the region of where I was interested in studying is the retrospinal cortex. And both these regions talk quite a bit to each other. So they're in constant communication, right? Because not one brain region is involved. In memory, lots of different brain regions come into play. So these two brain regions are chat with each other, coordinate and communicate with each other, just with those, just like with the electrical signals and action potential that I was talking to you about.
Great, thanks so much for showing that it's more than an angry face. It might look a little bit like an angry face here, but there's a lot going on there.
It was. I'm sorry. I should have. But I'm glad someone brought that question up. It can look like an angry face, but yep, the hippocampus is like the seahorse of the brain. It's legitimately called the seahorse.
Okay, Hanna. Well, thanks again so much for such a wonderful presentation. I'm sure we're all going to take what we've learned to train our pets or our siblings, as another volunteer alluded to, back when we get home. And so thanks again for teaching us so much today.
And thanks for all the great questions and your the exciting discussions as well.